Hey everybody, welcome to the last lecture in chapter 3. So we're going to talk about eukaryotic cells, and I'm going to probably go through this a little bit faster because most people have learned at least once in their life about eukaryotic cells. And so hopefully this is a refresher, but if not, I will give you information, and you can always find more information in your textbook if I didn't give you enough, okay? So we'll talk about eukaryotic cells. We'll focus on animal cells because that's what I typically talk about, but we'll talk about plant cells as well. And we'll look at the fact that eukaryotic cells are larger, more complex. They have more internal structures. The structures are known as organelles. This is an animal cell. This is a plant cell. What are organelles? I always liken organelles to the organs of our body. Organs have a specific function, and they do that for our bodies. Organelles have a specific function for a cell, okay? And these organelles are found within structures like vesicles, and they have different jobs depending on what they do. The plasma membrane of the eukaryotic cell is similar to prokaryotic cells. They have proteins that serve as receptors or serve to allow for materials to move across the membranes. Eukaryotic cells have sterols. In animals, this is cholesterol. In fungi, it's ergosterol. We did talk about mycoplasma, which has sterols as well, but they are not as common in your bacterial cells. The bacterial plasma membrane functions in producing ATP through the electron transport chain, but eukaryotic plasma membranes do not. So ATP synthesis occurs within the mitochondria of the cell. In general, prokaryotics and eukaryotic cells have similar transport mechanisms. Some differences, though, are the endocytosis and exocytosis, which are only found in eukaryotic cells. So endocytosis, we have three forms. Pinocytosis, which is basically cellular drinking. You're taking a tiny amount of fluid from the extracellular region. You form a small vesicle and pull that fluid in with any dissolved solutes. That's pinocytosis. Phagocytosis is used by some cell types. So pinocytosis in general can be used in all animal cells can use this, whereas phagocytosis is used by certain types of protozoans and certain types of animal cells. And phagocytosis is likened to eating, so cellular eating. You take in a large molecule, you might take in an entire bacterial cell or a damaged cell or a large amount of material. You form a vesicle with the plasma membrane around that material, pull it in, and then you fuse to a lysosome, and the lysosome can degrade the material. Receptor-mediated endocytosis is a specialized form of endocytosis in which certain molecules bind to ligands, and once all the ligands are bound, then all the material is pulled in. This is how animals get rid of excess cholesterol from the blood. And then we have exocytosis. Exocytosis is the release of materials from the cell by taking a vesicle, fusing that vesicle to the plasma membrane, opening the vesicle, and releasing that material out. So it's the reverse of endocytosis. Endocytosis is taking in materials into the cell. Exocytosis is material exits the cell, okay? Oh, shoot, sorry. All right, there we go. So now let's talk about ribosomes. I already mentioned ribosomes are the site of protein synthesis. Eukaryotic and prokaryotic ribosomes differ in size, and that differing in size is oftentimes what we target with antibiotics. If the bacteria cannot undergo protein synthesis, then they cannot produce proteins for replication, and they would die then. Eukaryotic cells have a cytoskeleton composed of 3 different protein types: actin filaments, microtubules, and intermediate filaments. Together, they form the cytoskeleton. The cytoskeleton is the framework of the cell. It also allows for movement of materials throughout the cell. Microtubules help in forming cilia and flagella, as well as the spindle fibers used to move DNA around during DNA replication and mitosis and meiosis. Flagella and cilia then are structures that help to either move the cell or move materials along the cell's surface. Flagella are much longer than cilia. In eukaryotes, they move materials in one direction. So they don't do that run and tumble. They just move forward, or the entire cell can turn and then they move away from something. Cilia are much shorter, and when they beat synchronously. They can move materials along the surface of the cell, or they can move the cell towards or away from something. The nucleus is then the cell's brain, basically the brain of the cell. This is where all the DNA is found. It also contains a nucleolus. The nucleolus is where ribosomes are formed. So the nucleus is where we hold all of our DNA and where RNA synthesis occurs. The nucleus has 2 membranes. It has a double membrane. It has these little nuclear pores, so materials can move in and out of that nucleus. But DNA cannot move out, or just out of the nucleus. It's already in there. The mitochondria is the site of ATP synthesis. We often call it the powerhouse of the cell. Mitochondria has 2 membranes, and we're going to talk about the mitochondria in the endosymbiotic theory. So something I always point out is that mitochondria contain their own DNA. The DNA is circular, and it's one single circular chromosome, just like bacterial DNA. Mitochondria have their own ribosomes. These ribosomes are 70S ribosomes, just like bacterial ribosomes, and they can replicate without the cell replicating. Chloroplasts are the site of photosynthesis. They're found in plants and algae. Chloroplasts contain a light absorbing molecule called chlorophyll. So they can take in sunlight energy, and then they use that to produce ATP. ATP then is used to convert carbon dioxide to a carbohydrate, sugar. Chloroplasts are also going to be discussed in the endosymbiotic theory. Chloroplasts also contain their own single circular strand of DNA, just like bacterial DNA. They also have their own 70S ribosomes. Both the mitochondria and the chloroplasts are double membrane bound, and they have their own proteins as well, and they can undergo replication without the cell. So what is the endosymbiotic theory? This is a theory that bacteria helped in producing the first organelles, some of the first organelles. So ancestors of mitochondria and chloroplasts were actually bacteria. There are a lot of evidences that support this, which I've talked about a little bit. The mitochondria and chloroplasts both have their own DNA, and the DNA is very similar to bacterial DNA. They contain their own 70S ribosomes. They have their own, they can undergo protein synthesis on their own because of this. They replicate via binary fission, very similar to, they replicate very similar to bacteria via binary fission. If we take out mitochondria or chloroplast from a cell, the cell cannot reproduce those structures. And they can replicate on their own, like inside of a cell. So they don't need the cell to tell them to replicate. So all of these are support for this theory. This actually became a theory in the, I believe it was 1967, and I will check just to make sure. So in the late 60s, this theory was proposed. Initially was a hypothesis back in the 1920s, and it wasn't a super popular theory, but finally in the 80s it became the best explanation to how eukaryotic cells were formed. And so we do support this theory now. I think all scientists believe the endosymbiotic theory as how eukaryotes evolved from prokaryotic structures. One last thing I will mention is that there are some cells that don't have or have very few, if any, mitochondria. These cells are always parasitic. So you're looking at organisms that cannot utilize, I'm not talking about viruses, I'm talking about eukaryotic cells that have no mitochondria, and these cells are going to be, if they have no mitochondria, then they're going to be obligate intracellular parasites because they have to use the energy made by another cell to produce their ATP to drive their own cell functions. The next organelle that we'll talk about is the endoplasmic reticulum. There are two types of endoplasmic reticulum. You can see them here. We have one that is dotted with ribosomes. So this is called the rough ER. And then out here we have the smooth ER. Rough ER synthesizes functions in protein synthesis. And these proteins are not destined for the cytoplasm. So we have ribosomes in the cytoplasm as well. But the ribosomes here are going to eventually move to the Golgi and then be shipped out. Smooth endoplasmic reticulum is the site of lipid synthesis and degradation of toxins and lipids. The Golgi then is a part of that endomembrane system that functions in taking materials from the ER and then shipping them to the areas that they need to go. Materials in the Golgi are going to be potentially processed or modified a little, packaged, and then sent out in little vesicles. Lysosomes are the digestive system of the cell. So lysosomes break down materials that have been taken in via endocytosis. Peroxisomes help to degrade lipids. They detoxify chemicals. They also can bind with vesicles and provide what we call an oxidative burst to kill the material, like bacteria. Peroxisomes contain specific enzymes that will degrade substances like hydrogen peroxide to oxygen and water to help protect our cells from superoxide, or not superoxide, from the free radicals of oxygen. Oh, that's the last slide. I'm sorry, I didn't realize I was on the last slide. I thought there was one more. So I'm going to stop here and I will post this video and I will start the next chapter in just a little bit. Have a great day. Bye.